Hello everyone. Today we will study about biomolecules. This is part 2. Let's start. Disclaimer. The images used in our slide deck are for educational purposes only. We respect the intellectual property rights of original creators and owners of these images. If you are the owner of any images used in this deck and have any concerns or issues, please contact us at sathee@iitk.ac.in and we shall remove the images. Some important topics which we will going to cover today is first one, polysaccharides, proteins, classification of proteins, structural level of proteins, enzymes, vitamins, nucleic acids, biological functions of nucleic acids. So let's study about our first topic, that is polysaccharides. Carbohydrates which yield a large number of monosaccharide units on hydrolysis are called polysaccharides. So carbohydrates which yield a large number of monosaccharide units when we do their hydrolysis are known as polysaccharides. For example, starch, cellulose, glycogen, gums arabic which is acidic, etc. So all these type of carbohydrates are polysaccharides. Polysaccharides are not sweet in taste and hence they are called non-sugars. Moreover, all polysaccharides are non-reducing due to the absence of free aldehyde CHO or carbonyl C double bond O group. Okay. So all polysaccharides are non-reducing due to the absence of free aldehyde or carbonyl group. Some of the important polysaccharides are as follows. First one is starch. Starch is the main storage polysaccharides of plants. So uh, in the plants, starch is present, which is the main storage polysaccharides in the plants. It is a polymer of alpha D glucose units. That means by joining various alpha D glucose units, we can form starch. So starch is a polymer of alpha D glucose units and consists of two components, amylose, which is water soluble, and amylopectin, which is water insoluble. So starch is a polymer of alpha D glucose and it consists of two components, amylose and amylopectin. One, the amylose is water soluble and the amylopectin is water insoluble. Second carbohydrate or polysaccharide is cellulose. Cellulose is a predominant constituent of cell wall of plant cells. So in the plant cells, the cell wall is situated, and in the cell wall we have a predominant constituent of cell wall which is cellulose. So cellulose is present in the cell wall of plant cells. It is a straight chain polysaccharides composed only of beta D glucose units which are joined together by beta 1,4 glycosidic linkage. So cellulose is a straight chain polysaccharides in which beta D glucose units are present, okay, which are joined together. uh, as I said that it is a straight chain, so beta D glucose units are joined together by some kind of linkage, right? So that linkage is beta 1,4 glycosidic linkage, that is between the carbon 1 of one glucose and carbon 4 of next glucose unit. So between the two beta D glucose units, the bond is formed between the first carbon and the fourth carbon of two different glucose units. Cellulose is not digestible by the humans, cellulose is not digestible. So if you intake cellulose, you cannot digest, your body cannot digest that due to the absence of enzyme cellulase in the digestive system. Why? Because in the human digestive system, the enzyme known as cellulase is absent. Okay, cellulase helps to dissolve or to digest the cellulose, and that is why, as the cellulase is not present in the human's body, in the human digestive system, that is why the cellulose is not digestible by the humans. The third important polysaccharides is glycogen, which is the carbohydrates stored in animal body, and it is a condensation polymer of alpha D glucose. When the body needs glucose, enzymes break the glycogen down to glucose. So glycogen is basically stored in the animal body, a kind of carbohydrate. And when the body needs glucose, whenever the body needs to get some glucose, the enzymes break the glycogen carbohydrate down to the glucose and supply it to the body. Okay, so these are three important polysaccharides: starch, cellulose, and glycogen. Now we will study about proteins. So what are proteins? Proteins are large biomolecules that occur in every living organism. So in the every living organism, proteins are situated as a large biomolecule. Proteins are polymers of alpha amino acid. What does that mean? That by joining the different amino acids, you can get protein structure, or you can say indirectly that proteins are the polymers of um, amino acids. Amino acids, compounds which have both the acid and the amino group, as the name says. So in the amino acids, both groups are present, amino and the acid group. So amino acids, first of all, the total number of amino acids that have been found in the proteins are 20. On the basis of their synthesis, that how they will form, amino acids are divided into two classes. First class is essential amino acids. Essential amino acids are the amino acids which cannot be synthesized in the body and must be obtained through diet. So, as the name says, essential amino acids, that it should be present in the body, right? Essential means important. But essential amino acids are the amino acids which cannot be synthesized in the body by itself, and it must be obtained, or it must be supplied into the body through the diet. Okay, so what are the example of these type of amino acids? Example are valine, leucine, lysine, isoleucine, or arginine, etc. All these are essential amino acids which must be taken or obtained through the diet and cannot be synthesized in the body by itself. The second important class of amino acids is non-essential amino acids. They are the amino acids which can be synthesized in the body. So they can be synthesized in the body by themselves. For example, glycine, alanine, glutamic acid, aspartic acid, etc. So all these are the example of non-essential amino acids. Okay, non-essential, as they can be synthesized in the body, you cannot or you should not have to obtain it from your diet. Amino acids behave like salts rather than simple amines or carboxylic acids. As we discussed that amino acids having both the group acid group and the amino group, so amino acids behaves like a salt and not like simple amines or any carboxylic acid. Okay, This is due to the presence of both acidic and basic groups in the same molecule, because amino acids is having both group, acid group and amino group. That means acidic group and basic group in the same molecule. That is why it behaves like a salt rather than simple amines or carboxylic acids. This is the structure of a amino acid, general structure: R C H NH2 COOH. So you can see that we have our acid group, we have our amino group attached to the or present in the same molecule, and this is a amino acid. It can be interconvertible to RCHCOO negative, and on the C we have NH3 positive. So you can see that negative and positive charge both are present in the same molecule. Negative by the acid group, positive by the amino group. And this structure is known as Zwitter ion. Okay, because both the negative and the positive charge is present in the same molecule in the same structure. So this is known as Zwitter ion. At a certain pH of the medium, called the isoelectronic point of an amino acid, the structure behave as a dipolar ion or Zwitter ion. So the medium in which we have stored the amino acids, or in which our amino acid is present, the medium or the at a certain pH of that medium, which is called that certain pH is called isoelectric point of an amino acid. So at that point, the structure of the amino acid behave as a dipolar ion, or you can say Zwitter ion, and does not migrate to any electrode on passing current, and it will not migrate to any of the electrode, either cathode or anode, when we pass current. Why? Because both type of charge will be present in the Zwitter ion, so it will not migrate to any type of electrode both. Okay, when we pass current into the medium. Proteins give Burette test, Millions test, or 9-hydrin test. So these are the tests which is given by the proteins. Peptides and peptide linkage. So what are peptides and what is peptide linkage? The basic structural unit of protein is alpha amino acids, because protein are the polymer of alpha amino acid. That means the structural unit of proteins through or by which protein is made up of is alpha amino acids. Amino acids may be joined together by an amide linkage called peptide linkage. So as various structural unit of alpha amino acids, basically various units of alpha amino acids are present and joined together to form a protein. So what kind of bond is that? So they will be joined together by the amide linkage, which is known as peptide linkage. So that amide linkage is called peptide linkage, basically, like, okay. CO NH, this bond between CO and NH is known as peptide linkage, which is formed between the two amino acids. For example, NH2CH2COH, which is a amino acid, plus NH2CH3COH. Okay, so this is another kind of amino acid. So there are two amino acids. When you combine them, when you join them, so one molecule of water will be lost, and there will be formation of peptide linkage. Okay, so it will become NH2CH2CO NHCH COH CH3. Okay, and the CONH bond is known as peptide linkage. So, and we will get a diapeptide molecule by the combination of two alpha amino acids, and when they lost a water molecule, they form peptide linkage and we get a diapeptide molecule. The molecule derived from two amino acids containing a single peptide linkage is called diapeptide. So a molecule uh, which is derived from two amino acids. Okay, like two amino acids combined together to form that molecule, and that molecule containing a single peptide linkage. So that is known as diapeptide. That molecule is known as diapeptide. That derived from three amino acids. If a molecule derived from three amino acids, that means three amino acids combined together to form a molecule, then that molecule is termed as tripeptide linkage. Sorry, tripeptide. Okay. The peptides having 2 to 10 amino acids residues are called oligopeptides. So any peptide in which the structural units are 2 to 10 number of amino acids, they will known as oligopeptides, while those with the greater than 10 amino acid residues or their structural units will be more than 10 amino acids, they will be called polypeptides. Poly means more than or many. Okay, so in the case of proteins, it will be more than 10. Polypeptide with the molecular weight greater than 10,000 unit is termed as protein. So any protein, uh, you can term the polypeptide with the molecular weight greater than 10,000 unit. You can term that as a protein. Classification of proteins. Proteins can be classified into two types on the basis of their molecular shape. Depending on the molecular shape of the protein, we can classify the protein in the two basic types. First one is fibrous proteins. Polypeptide chains form fiber-like structure, for example, keratin and myosin, etc. So keratin and myosin are fibrous proteins. They form a fiber-like structure. Second one is globular proteins, and this structure results when the chain of polypeptide coils around to give a spherical shape. So fibrous protein contain fiber-like structure, and globular proteins become the type of structure when the chains of polypeptides coil together to give us a spherical shape. Then we can get globular protein, and these are usually soluble in water. For example, insulin and albumins. These are the example of globular proteins. Structural levels of proteins. So on the structural level of proteins, how can we see that? Structure and shape of proteins are of four different levels. So if you see at the structure and the shape of the proteins situated to the protein, they are of four different levels. They are primary structure, which involves polypeptide that has amino acids linked in a specific sequence. Change in this structure gives a different protein. So basically, in the primary structure of proteins, the chains or the amino acids uh, in the polypeptide is linked in a specific sequence. If you change that sequence, if you change that structure, you will get a different protein. So that is known as primary structure. Second one is secondary structure. So secondary structure of proteins involve linking of polypeptide chains by the hydrogen bonds with the help of hydrogen bonds. They are found to exist in two different types of structures, alpha helix and beta plated sheet structure. So secondary structure proteins are further can be found into two different types, alpha helix and beta plated sheet. Then we have tertiary structure of proteins, represents overall folding of the polypeptide chains, and it involves the polypeptide bonds, hydrogen bonds, disulfide linkages, van der Waals forces, and electrostatic force of interaction. So in the tertiary structure we have everything: polypeptide bond, hydrogen bond, disulfide linkage, van der Waals forces, electrostatic forces of attraction. It gave rise to two major molecular shapes, fibrous and globular. So tertiary structure gave rise to two main structure or shapes of proteins, which is fibrous and globular. Fourth one is quaternary structure. So quaternary structure of proteins composed of two or more polypeptide chains referred to as subunits. So in the quaternary structure, there can be two or more polypeptide chains which you can refer as subunits. The spatial arrangement of these subunits, when you arrange these subunits spatially with respect to each other, they become or they form the quaternary structure of the protein. Denaturation of proteins. What will happen if you do the denaturation of proteins? Disturbance of hydrogen bonds through which the polypeptide chains are holding together, either by acids or alcohols or heat. So when you disturb the hydrogen bonds, either by the acids or the alcohols or heat, results in the unfolding of globules. So what will happen? The globules will get unfolded and thus helix get uncoiled, and the protein loses its biological activity due to the change in the temperature or the pH, because so many changes happening. Hydrogen bond will get disturbed, the coil change will get uncoiled, the folding gets unfold. So due to the then temperature and pH will also change. So due to that, the protein will lose its biological activity. During the denaturation, secondary and the tertiary structures of proteins are destroyed while the primary structures remain intact. Primary structures remain as it is, while the secondary and tertiary structures destroyed while the denaturation is happening. Let's see now about the enzymes in details. So enzymes are globular proteins which are biological catalysts. You can say enzymes are catalysts biologically. Enzyme inhibitors reduce the activity of a particular enzyme. Enzyme inhibitors are what? They reduce the activity of a particular enzyme. These are mostly inorganic ions or complex organic molecules. Almost all the enzymes are globular proteins. These are very specific for a particular reaction and for a particular substrate. So enzymes are very specific for what the work has been assigned to them. What will be their role? So what will be the particular reaction for which they have to enter, or for the particular substrate, okay, to which they have to attached. So almost all the enzymes are very specific for that. Congenital and albinism disease are caused by the deficiency of enzymes phenylketonuria and tyrosinase respectively. So if you lack these type of enzymes, they can also cause you uh, in any kind of disease. For example, if you have the lack of enzyme phenylketonuria, you can get congenital disease, or if you lack of the enzyme tyrosinase, then you can get albinism disease. Let's see about the vitamins. Organic compounds, they are organic compounds required in the diet in small amounts, not in the large amount. In the small amount, we need it in our diet to perform specific biological functions for the normal maintenance of optimum growth and health of the organism. So for any organism, their growth and their health is important, right? And for their health and for their optimum growth, uh, in their diet, organic compound is required to perform specific biological functions, which are termed as vitamins. So these organic compounds are vitamins. Provitamins are the biologically inactive compounds that have almost same structure as the vitamins. There is another term, provitamins, which are structurally same as vitamins. They are the biological inactive compounds and they can be converted easily into the active vitamins. For example, beta carotene is a provitamin for the vitamin A. That means beta carotene is an inactive form. It can be converted easily to the active vitamin, which is vitamin A. Various types of vitamins are as follows. Fat or oil soluble vitamins. So first one is fat or oil soluble vitamins. For example, vitamin A, D, E and K. Second one is water soluble vitamins. For example, B group vitamins like vitamin B1, B12 and vitamin C. So they are water soluble, that can be soluble in water but not in fat. And the fat one are that can be soluble in the fat but not in the water. Some important vitamins, their sources and their deficiency diseases are. So we have a table in which 3 columns are present. First one is name of the vitamin, second one is about sources, and the third one is about deficiency disease. So vitamin name is vitamin A, retinol. The sources of vitamin A is fish liver oil, carrots, butter and milk. So if you have a deficiency of vitamin A, it will cause you it will cause you xerophthalmia disease, which is hardening of cornea of eye and night blindness. Second vitamin is vitamin B1, which is from the group B vitamin, thiamine. The sources of this vitamin is yeast, milk, green vegetables, and the deficiency of vitamin B1 can cause you beriberi disease, which is loss of appetite. Third one is vitamin B2, riboflavin. The sources of vitamin B2 is milk, egg white, liver, kidney. And the deficiency of vitamin B2 can cause you chlosis disease, which is fissuring at corners of mouth and lips. Fourth one is vitamin B, pyridoxine. Sources of vitamin B is yeast, milk, egg yolk, egg yolk, cereals. The deficiency of vitamin B can cause you convulsions and nervousness. Important vitamins, their source and their deficiency disease table continued. So next vitamin is vitamin B12, cyanocobalamin. The sources of vitamin B12 is meat, fish, egg and curd. The deficiency of vitamin B12 can cause pernicious anemia, which is RBC deficiency in hemoglobin. Next vitamin is vitamin C, ascorbic acid. The sources of vitamin C is citrus fruit, amla, and green leafy vegetables. The deficiency of vitamin C can cause scurvy, which is bleeding gums. Next vitamin we have is vitamin D. The sources of the vitamin D is exposure to sunlight, fish, and egg yolk. The deficiency of vitamin D can cause you rickets and osteomalacia. Next vitamin is vitamin E. The sources of vitamin E is wheat, germ oil, sunflower oil, and the deficiency of vitamin E can cause increased fragility of RBC and muscular weakness. Then we have our vitamin K. The most sources of vitamin K is green leafy vegetables, and the deficiency of vitamin K can cause increased blood clotting time. Let's see now about nucleic acids. These are responsible in the biosynthesis of proteins. Nucleic acids are biological polymers. They function as the chemical carrier of cells genetic information. So they carries chemically the cells genetic information. These are mainly of two types: deoxyribonucleic acid, which is DNA, and ribonucleic acid, which is RNA. First one, DNA, deoxyribonucleic acid. DNA is the polymer of nucleotide. That means when you join nucleotide, many, you can get DNA polymer. and it is a genetic material related to the genetic information. It has double helical structure. Nucleotide has deoxyribose sugar, phosphate bond, and nitrogenous base. Nucleoside has deoxyribose sugar and nitrogenous base. So in the nucleoside there is absence of phosphate, a unit formed by the attachment of base to a to a first position of sugar is known as nucleoside. So when a base is attached to the first position of sugar, a nitrogenous base is attached at the first position of deoxyribose sugar, you can get a nucleoside. So in the nucleoside there is no phosphate present, but as a nucleotide there is three deoxyribose sugar, phosphate, and nitrogen base. All three are present. In nucleosides, the sugar carbons are numbered as 1 prime, 2 prime, 3 prime, etc. in order to distinguish these from the bases carbons. Okay. When nucleoside is linked to the phosphoric acid at 5 prime position of sugar moiety, we get a nucleotide. So from the nucleoside we get nucleotide. How? When nucleoside is linked to the phosphoric acid at the 5 prime position of the sugar moiety. Okay, as the sugar carbon are named as 1 prime, 2 prime, 3 prime. So on the 5 prime position of sugar moiety, when you linked the when you link the nucleoside, okay, to the phosphoric acid at 5 prime position of sugar moiety, then we can get a nucleotide. Nucleotides are joined together by phosphodiester linkage between 5 prime and 3 prime carbon. DNA contains 4 bases: adenine, guanine, cytosine, and thymine, and their respective symbols are A, G, C, and T. Here, adenine and guanine are called purines, whereas thymine and cytosine are called pyrimidine. DNA has A equivalent to T and C, that is cytosine, equivalent to G, that is guanine. So their structures are equivalent. Adenine equivalent to thymine, cytosine equivalent to guanine. Second nucleic acid is RNA, that is ribonucleic acid. It is also a polymer of nucleotide units, but here the nucleotide unit contains ribose sugar instead of deoxyribose. RNA has adenine, guanine, cytosine, and uracil instead of thymine. So in the RNA, uracil base is present instead of the thymine, as thymine is present in the DNA along with adenine, guanine, and cytosine. RNA molecules are of three types. These are messenger RNA, mRNA we can say, ribosomal RNA, rRNA, and transfer RNA or tRNA. They perform different functions. As they have different name, they perform different functions in our body. Biological functions of nucleic acid. So we are uh, studied that the nucleic acid is related to the biological function, but what are they? So DNA has an ability of self-duplication during cell division, and identical DNA strands are transferred to the daughter cell. So basically when a cell is divided into two, DNA has the ability of self-duplication, and the daughter cell, okay, so identical DNA strand, so basically DNA will get self-duplicate and that identical DNA strand will get transferred to the daughter cell, to the new cell which just divided from the parent cell. In this way, DNA is responsible for maintaining the identity of different species of organism over million of years. And as DNA can get self-duplicate, and transfer its strand, identical strand from one cell to the another cell, from parent cell to the daughter cell. And this is the mechanism, or I can say this is the way through which DNA is responsible for maintaining the identity of different species of organism for the millions of years. RNA molecules synthesized various types of proteins in the cell, but the message for the synthesis of a particular type of protein is present in the DNA. So the work of RNA molecules is to synthesize the various types of proteins, but in the cell. Okay, but this message that RNA have to synthesize the proteins, this message is synthesized, or this message for synthesis of particular type of protein is present in the DNA. Genetic messages are encoded in DNA, while RNA translated the encoded messages from DNA and thus helps in the protein formation. So all the genetic information in the coded form is present in the DNA, and the RNA work is to translate that encoded message from DNA and helps in the protein formation. Good evening and hello everyone. Today we will going to see some practice questions from biomolecules, and this is part 2. Let's start. So today we will see some practice questions from question number 9 to question number 16. Let's start with the question number 9. Which of the following pairs give positive tolerance test? We have given some pairs in the options. We have to tell that which pair give positive tolerance test, means which pair, in which pair both the components will pass the tolerance test. Options are glucose and sucrose, glucose and fructose, hexanol and acetophenone, or fructose and sucrose. You have 10 seconds to think about this question. After that, we will see the answer. The correct answer for the given question is option B, that is glucose and fructose. How? Glucose and fructose give positive tolerance test as they are reducing sugar because of the presence of CHO group or free CHO group. So glucose and fructose both will pass the tolerance test. Why? Tolerance test will be for the presence of the aldehyde group or carbonyl group. Okay, and because in the glucose and the fructose structure we have the presence of free CHO group or free aldehyde group, carbonyl group, that is why both glucose and fructose pass the tolerance test, gives positive tolerance test, and they both are the reducing sugar, and due to the presence of free CHO group. That is the main reason, and that is why we have our correct answer as option B, that is glucose and fructose. The beta and alpha glucose have different specific rotations. When either is dissolved in water, their rotation changes until the same fixed values results. This is called what? So, we have two forms of glucose, beta glucose, alpha glucose. We have asked that the beta glucose and alpha glucose have different specific rotation. Their rotation will also be different and specific, the alpha 1 and beta 1. And we have asked that when either one is dissolved in the water, either alpha or beta, either one is dissolved in the water, their rotation, their specific different rotations changes until the same fixed value results. The value which is fixed for the particular, you know, uh, glucose like beta glucose or alpha glucose, until that fixed value results, the rotation will keep on changing when we dissolve our alpha glucose or beta glucose in the water. So what this is called? Options is epimerization, racemization, anomerization, or mutarotation. You have 10 seconds to think about this question. After that, we will see the answer. So the correct answer for the given question is option D, that is mutarotation. The beta and alpha glucose have different specific rotations. When either is dissolved in water, their rotation changes until the same fixed value results, and this phenomena is called mutarotation. In the mutarotation are the product or the compound having different specific rotation will keep on changing until the fixed value results when the product is dissolved in water. So basically this is mutarotation, and this is the concept which is shown by the beta and alpha glucose when they dissolved in water, and that is why our correct answer is option D, that is mutarotation. Which of the following statement is not true about glucose? So we have to tell that from the given options which statement is not true or is false about the glucose. Options are: it is an aldohexose. On heating with hydrogen iodide, it forms an hexane. It is present in furanose form, or it does not give 2,4-DNP test. You have to tell which statement is not true regarding glucose. You have 10 seconds to think about this question. After that, we will see the answer. So the correct answer for the given question is option C, that is, it is present in furanose form. The cyclic structure of glucose is represented by Haworth projection as a pyranose ring and not in the furanose form. So basically we have a cyclic structure of glucose. So glucose is having cyclic structure which is represented by Haworth projection as a pyranose ring, not in furanose form. Pyranose derived from word pyrene, which means six-membered cyclic ether. Okay, so basically, pyranose we know from the word pyrene, which means six-membered cyclic ether, and the cyclic structure of glucose related with the pyranose because it is represented by the Howard projection as a pyranose ring, like six-member ring, and not in the furanose form, and that is why uh, the statement the statement given in the option C, that it is present in furanose form about the glucose, is not true, and this is our correct answer. So option C is the correct answer. The term invert sugar refers to an equimolar mixture of what? So we used invert sugar term. So we have asked that the term invert sugar refers to the equimolar, that means equal number of moles, equimolar mixture of what? Which two compounds? Options are D-glucose and D-galactose, D-glucose and D-fructose, D-glucose and D-mannose, or D-glucose and D-ribose. You have 10 seconds to think about this question. After that, we will see the answer. Okay, so let's see. The correct answer for the given question is option B, that is D-glucose and D-fructose. D positive sucrose, which is called as invert sugar, is an equimolar mixture of D positive glucose and D negative fructose. So D positive sucrose, known as invert sugar, is a mixture of D positive glucose and D negative fructose. It is called invert sugar due to the fact that on hydrolysis there is a change in the sign of the rotation from positive to the negative. That is why it is called invert sugar. Because when we do the hydrolysis for the D positive sucrose, it will change their sign of rotation from the positive to the negative, right? And that is why the question we have asked, that invert sugar term refers to the equimolar mixture of what? So the correct answer is option B, that means equimolar mixture of D-glucose and D-fructose. These are the two components in the equimolar amounts present in the invert sugar. Burette test is not given by, select from the options. Options are carbohydrates, polypeptides, urea, or proteins. You have 10 seconds to think about this question, and then we will see the correct answer. Okay, let's see. So the correct answer for the given question is option A, that is carbohydrates. All the compounds containing NHCO bond, that is peptide bond, give Burette test. So Burette test will be positive for all the compounds containing the peptide bonds. Since carbohydrates do not contain the peptide bond, it does not give the test, and carbohydrates do not contain any kind of peptide bond, that is why they will not give the test of Burette test, right? So this is the question, that Burette test is not given by which product, so they are called carbohydrates, and thus our correct answer is option A, that is carbohydrates. Which one of the following statement is correct? We have to tell the correct statement from the options. Options are all amino acids except lysine are optically active. All amino acids are optically active. all amino acids except glycine are optically active, or all amino acids except glutamic acids are optically active. You have to choose the correct statement. You have 10 seconds to think about this question. After that, we will see the answer. Okay, let's see. So the correct answer for the given question is option C, that is all amino acids except glycine are optically active. That means every amino acids except the glycine amino acid, every amino acid is optically active. The correct statement is that all amino acids except glycine are optically active. This is the concept. This is the correct concept. In the glycine, there is no chiral carbon. And as there is no chiral carbon in the glycine, that is why it is optically inactive. Because in the glycine, if the chiral carbon is absent, then there is some kind of symmetry present in the glycine. That is why it is optically inactive. And other than that, every amino acids except glycine is optically active. So our correct statement will be option number C, that is all amino acids except glycine are optically active. Okay, now let's move on to our next question. The bond that determines the secondary structure of protein is. Which bond determines the secondary structure of protein? Options is coordinate bond, covalent bond, hydrogen bond, or ionic bond. You have 10 seconds to think about this question. After that, we will see the answer. So the correct answer for the given question is option number C, that is hydrogen bond. Secondary structure of protein is refers to the shape in which long polypeptide chains are held together by the means of hydrogen bonding. So that is the correct answer, that the secondary structure of proteins refers to the shape shape in which the long polypeptide chains are held together by the means of hydrogen bonding. Between them, strong hydrogen bonding is present. That is why the long polypeptide chains held together in the shape of the protein in the secondary structure. So, which bond determines the secondary structure of the protein? Correct answer is option C, that is hydrogen bond. Let's move on to the next question. Which of the following compounds can be detected by Molish test? Which compound can be detected by Molish test from the options? Options are nitro compounds, sugars, amines, primary alcohols. The correct answer for the given question is option number B, that is sugars. Carbohydrates, for example sugar, can be detected by Molish test, and the test sample, add the test sample to the test tube containing water. That means we have to take a test tube having water in it, and we have to add the unknown sample in it and shake it so that it will get dissolved. Now we have to add 2 to 3 drops of Molish reagent and add concentrated sulfuric acid in that in drops along the sides of the test tube slowly. The formation of a purple ring indicates the presence of carbohydrates. If a purple ring formed in that particular test tube, we can say that yes, the carbohydrate is present in our test sample. Okay, so we have asked that which compound can be detected by Molish test. So correct answer is option B, that is sugars. Okay.